Hello everyone, my name is Eduardo Lamas, and in this video we are going to talk about pattern recognition receptors or PRR. PRRs are a primitive part of the immune system. They identify self or host proteins from non-self or pathogens. PRRs are proteins expressed by cells of the innate immune system. PRRs identify two classes of molecules: pathogen-associated molecular patterns or PAMPs, which are associated with microbial pathogens, and damage-associated molecular patterns or DAMPs, which are released during cell damage or death. PRRs are non-specific as compared to antibody recognition, and the response is rapid. PRRs are able to recognize structures shared by a class of microbes or molecular patterns. As shown here, different pathogens may share molecular patterns or PAMPs. Many examples of common patterns are available. For instance, mannose patterns are found on different types of organisms. LPS is found on gram-negative bacteria, and LTA is found in gram-positive bacteria. These PAMPs are recognized by PRRs, which are found on macrophages. Here, for example, we have a PRR called TLR or toll-like receptor. On the other hand, in the case of the adaptive immunity, pathogens are recognized specifically as is shown here. Each pathogen is specifically recognized by a single antibody that is produced by a single clone of B cells. As you see here, antibodies are able to discriminate each pathogen, as each antibody is able to identify a specific structural detail of microbial molecules, also called antigens. Antibodies are able to detect minor differences found on pathogens. Therefore, PRRs are specific because they recognize pathogens, but not specific when compared to the adaptive immunity. The location of PRRs indicates where the infection is taking place. PRRs expressed on the cell membrane, outside of cells, allow recognition of PAMPs found on the surface of pathogens. Here, TLRs are well characterized PRRs, with TLR 1, 2, 4, 5, and 6 recognizing patterns of extracellular pathogens. PRRs are also found in vesicle compartments like the endosome. In this case, the pathogens are phagocytosed and recognized by PRRs inside the vesicles. Examples of these are TLR 3, 7, 8, and 9, which recognize nucleic acids found in the pathogens. PRRs are also found in the cytosol, and they include RIG, which recognizes viral pathogens, and NOT, that recognizes bacterial pathogens. These pathogens are intracellular and they manage to escape other PRRs, as well as infect the cells in the cytosol. PRRs are expressed in many innate cells, but its expression varies depending on the cells. In addition, each PRR recognizes different PAMPs. For instance, toll-like receptor 2, which is associated with TLR 1 and 6, is found mainly in macrophages, mast cells, and dendritic cells. These cells are found in intact tissue ready to recognize pathogens. TLR2 recognizes LTA, or lipoteichoic acid, which is present on gram-positive organisms. TLR3 is an intracellular PRR that recognizes the RNA of double-stranded RNA viruses. TLR3 is commonly found in natural killer cells, which are great in recognizing viruses and releasing cytokines against those viruses. TLR4, with the help of MD2 and CD14, recognizes LPS on gram-negative bacteria. TLR4 is mainly found on macrophages, but can also be found in dendritic cells, mast cells, and eosinophils. TLR5 mainly recognizes flagellin, which is present in certain bacteria such as Salmonella. The main cellular distribution is the intestinal epithelium. TLR7 8, and 9 are also intracellular and recognize intracellular pathogens, such as single-stranded RNA viruses. TLR 7 and 8 both recognize single-stranded RNA viruses, but TLR 8 are mainly found in natural killer cells, whereas TLR 7 can also be found in B cells, plasmacytoid dendritic cells, and eosinophils. TLR 9 recognizes DNA with unmethylated CpGs, which can be found in bacteria as some viruses. Recognition by PRRs and combination of different PAMPs tells innate cells the type of infection. For example, viruses have different PAMPs, which include glycoproteins, DNA, and RNA, and they can be recognized by different TLRs or proteins like RIG. Similarly, gram-positive bacteria have different PAMPs, such as lipoproteins, lipoteichoic acid. These are recognized by other TLRs and molecules of the immune system. The DNA of gram-positive and negative bacteria is recognized by TLR 9 and NALP3. Some PAMPs associated to gram-negative bacteria include LPS, flagellin, which are also recognized by different TLRs and molecules. More specifically, flagellin is recognized by TLR 5, LPS by TLR 4. Fungi also have their own PAMPs, and they include beta-glycans, mannans, and zymosan, which are recognized by different TLRs as well. As you can see, every organism has different PAMPs that can be recognized by the immune system. Recognition of PRRs and the combination of these different PAMPs can tell your immune system what type of infection you have. Engagement of the PRR pathway activates innate cells and stimulates cytokine production. When PRRs recognize PAMPs, they can lead to phagocytosis and activation of cells. In this example, different TLRs found on the surface of innate cells or in endosomes will lead to the recruitment of adapter proteins, the recruitment and activation of protein kinases, and the activation of transcriptional factors such as NF-kappa beta or IRF3, leading to gene transcription. In many instances, the engagement of PRR leads to the production of pro-inflammatory cytokines and chemokines. After watching this video, please rate it by using the thumb up or down option. Thank you.